Welcome to CEC Gurukul lectures on bioinformatics. Today we will be talking about the metagenomic data analysis. So for doing this, today's talk we will discuss this under 4 subheadings. Like first we will talk about what is metagenomics, meaning we will understand the boundaries and what all we study in metagenomics. Then we will know what sort of data we expect in metagenomics and how do we analyze this data. We will understand the bioinformatic workflow for analyzing this data, and we will also just touch upon the topic of machine learning, that how machine learning methods have been helpful in analyzing this metagenomic data. So starting first with the metagenome. And before we talk about metagenome, or maybe we first need to define microbiome. What is a microbiome? So microbiome refers to an entire microhabitat including its microorganisms, their genomes and the surrounding environment. So I said metagenome and then I said microbiome. So let's understand the difference between 3 terms. One is microbiome, one is microbiota, and we have metagenome. How these 3 terms are different from each other. Microbiome, the definition in biology, it refers to the microorganisms and their genes. So in microbiome we include the genes, whereas microbiota refers to the microbes themselves, meaning we just talk of microbes in microbiota. And if we just want to talk about the genes in the environment, we call them as metagenome. So the more meaning in metagenome, we don't take into the consideration the microbe from where they are coming. So microbiome lies between the microbiota and metagenome. So we try to identify the microorganism associated in microbiome, but we are hardly concerned with that when we talk about metagenomic studies. So before, for clear understanding of these 3 terms was quite important for analysis or for understanding the metagenomic data. Next is, here is one example, like what sort of data, where do we derive from in metagenomic data. So microbiome not only refers to the microorganisms, I told you already, but it also includes the environment. So it allows us to study the activity, meaning theater of activity, their interaction among each other. So this is a plant. I have taken this picture from the journal Nature, and here they show the different microbiomes a plant could have and how the microorganisms there in different locations, they vary in this pie chart. You can see that they vary in their abundance and diversity, and that basically leads to the interactions. So in microbiome, not only we talk about the diversity of the microorganisms, but about their interaction, their dynamic nature, and that leads to a better understanding of the ecosystem. And we also talk about the change with reference to time and scale. This is another example of a microbiome. Here I have just picked up a human microbiome because these studies initiated with human microbiome, and it has been applied extensively in humans. And as you can see in the drawing, we have microbiome isolated from different regions, maybe mouth or intestine, and they have different composition of microbiomes. And in human, in microbiomes, in the microbiome, or in a metagenome, the focus is on the environment, and the samples from different environments, as I said, maybe intestine or a plant rhizosphere or phyllosphere, or maybe mouth cavity, soil, the samples are taken from different environments. So environment is more important in these studies, and it can be it is it is being treated as a different unit for evolution, meaning it is now understood that all organisms are associated with different microbes together, and they together form another unit of evolution that is called as superorganism or meta-organism. So this is under debate, is that should we consider one organism individual, or we should also consider the associated microorganisms with that. So it's a point of discussion among the scientists. And now we come to Jo Handelman. She was the one who coined this term metagenome. So the metagenome, like she said, like we apply the genetic composition of one individual, we call that as genome. And when we study this genome from one environment altogether, the composition of that environment, we will call that as metagenome. So she is credited with coining this term metagenome, and her paper, she published this in her paper, and she is said to be she is given this contribution, and this article of hers was published in Pioneers in Science. So she coined the term metagenome, and metagenomics is application of modern genomic techniques to study the communities of microbial organisms directly in their natural environments, bypassing the need for isolation and lab cultivation of individual species. So this, with experience also, I used to work with rhizosphere microorganisms the long time back in 1988. How we used to study this is we used to isolate the microorganisms, culture them. It was a very long and a tedious process, and we could not get these several types of information from there. But in metagenomics, we also know the composition of the community which makes up this rhizosphere or any other microbiome. We can also know how they are interacting with the environment and what is the function which they are performing. So in metagenomics, we try to explore a bigger picture, meaning what is the relationship between the microbes and their habitat. In the bigger picture, we need a lot of experimental and computational techniques to be applied here, and this includes, we need to have information, and we can make inferences about marker genes, metagenomes, transcriptomes, metaproteomes, metabolomes. So like genomic studies, we use, when we take environment as one unit rather than an organism, we can isolate all the genes from there and we call them by different names. But that has been used to get amazing information from the data, like I tell you here in one example, where microbiome data from humans, and it has been used to study the antimicrobial resistance in elderly. Meaning how it was studied is that it was realized after this analysis of the microbiome that like some antibiotic like cephalosporin, it is highly variable. Some are susceptible to this, some are resistant to this. But if we look at erythromycin on the other hand, it has very low variation, meaning most of them are resistant to this. Same way, it can be used to decipher the information about, how that system or that person will respond. Meaning, if we compare this with the microbiome of a healthy person to that of a sick person, we can predict what response we expect, meaning which microorganisms will be killed and how it is going to affect. This can be done with this one, and it has really boosted the medicine studies with this one. Similarly, in an ecosystem, in an environment, in a forest ecosystem, maybe we could relate with several parameters like pH. The phylotypes and soil pH has been linked in other studies. Similarly, we could use this to correlate. This was an interesting study where kissing frequency was linked to the salivary microbiome, and it was observed that after up to 5-6 hours, the microbiome in the saliva, it stabilizes itself, and it is linked to the to the kissing frequency. So we can go to the all details of any phenomenon happening in a microbiome with metagenomic data analysis. So what we do in metagenomic process or the data analysis is we try to isolate. This is a marine ecosystem where we have a lot of microorganisms occurring. So rather than actually isolating the microorganisms, we can know with metagenomic analysis that what makes up this ecosystem, meaning which all organisms are existing there. We can get a very good idea and how they are interacting and what is the product of their interaction. So the broad applications of metagenomics would reveal the information about three aspects of an ecosystem. One is the biodiversity, meaning what microorganisms exist there. It would also tell us about the interactions, meaning how they are interacting with each other, and the phenotyping, meaning the manifestation of these interactions, meaning what character does come out as a phenotype as a result of these interactions. So that was about it. I hope I could convince you that metagenomics is a brilliant science which has come up after efforts, because for the scientists who've been working on these microbes in the rhizosphere or intestine, it has come up as a boom, and it really has helped in the pursuing the medicine and the other aspects of ecosystems. So going to the next subheading, that is what is the form of metagenomic data and how do we analyze that data. So for that one is first we need to understand, like the genomic studies, how do we extract this data. This data for metagenomic analysis can be a single gene survey, that is an amplicon. We usually amplify selected genes. It may be whole genome data, meaning the whole genome sequences are assembled. It may be meta-transcriptome, meaning the mRNAs from that particular environment, all mRNAs are isolated and assembled. It may be meta-proteome also, so that we talk about all the proteins in that system. So in the sequence-based metagenomics, we isolate the sequences, and what we do is, like in 16S rDNA sequencing, we select maybe 16S or 18S depending upon it is a prokaryote or it's a eukaryote. We will isolate the DNA from the samples, and then we amplify, sequence it, and then we do the phylogenetic analysis, and after that we compare these sequences. Same way in whole genome sequencing, we will do the sequencing and then we will assemble, we try to find out the genes and then we compare. The two data sets will be different and will have different applications and different implications in the metagenomic data analysis. So let's look at them little more details. Like, you know, in single gene or amplicon sequencing, we usually use 16S ribosomal DNA for prokaryotes, and we use 18S rDNA and ITS sequences for eukaryotes. The amplicon sequencing can be applied to low biomass specimens or samples, and which are usually, you know, they get contaminated by host DNA. So there it is good to use 16S sequences. But with this technique, we can identify the sequences only up to the genus level. But if we go for whole genome metagenomic studies, it is quite expensive as compared to single gene, and it gives you huge output in the form of up to 300 GB data. But here we get higher taxonomic resolution, and we can talk of the functional information also with whole genome metagenomic studies. So if we compare the two types of data obtained or two types of studies in metagenomic profiling, if we do marker gene profiling, that is single gene 16S, I said it's much less expensive, up to $100, meaning one sample can be analyzed. But for shotgun metagenomic profiling, it requires 10 times more dollars. It requires $1,000, and the computational needs are extensive for shotgun metagenomics. But in marker gene profiling, we could do this on a laptop or a small server. Then the the marker gene profiling could give you information about the taxonomic profiling, and it is, but in shotgun metagenomics it provides both taxonomic and functional profiling. We have lot of wasted data in shotgun metagenomics, but it is precise, but we could not identify. We can identify only up to genus or phylum level, but there is no waste data in marker gene profiling. Coming to meta-transcriptoming, in meta-transcriptomics we sequence the mRNAs of the whole community, and we talk of gene expression levels in that community, and it gives a very good snapshot of functional exploration of a microbial community in situ. Same way for metaproteomic data, we isolate all proteins from that environmental sample. We try to analyze them. It gives a good information about functional diversity and the structure of the microbiota in that particular niche, and it will also provide the information. Together with metabolomics, it can sort out lots of questions in the study. So here I was just trying to tell you that metagenomics was a very powerful technique and method, and it has been impacted by other studies also. Like if you see the cost, when the cost of sequencing came down, the same way it gives a multiplication of the papers on metagenomics and the unidentified proteins also. That number also increased tremendously. So going to what we do is, now I'll quickly introduce how the data analysis in bioinformatic data analysis workflow, meaning which steps we follow in there and what are the two platforms for doing this. And then in the next section we'll take that in details. So the workflow is that we have sequences, we input these sequences, and then we process these sequences, and we try to identify OTUs in that. We get a OTU table, and then in the downstream analysis we will analyze this data and we get a lot of information. We get graphics prepared with that information. We can also do the same thing with phylogeny and sequence alignment, drawing a phylogenetic tree. We have two platforms where the studies can be performed very efficiently. One is CHIME and the other one is Mothur. In the CHIME, it is a Python based platform, and it is more advanced than Mothur. And in CHIME, we have a wrapper sort of thing where all existing programs are existing, but Mothur on the other hand is re-implementation of the popular algorithms. Mothur can be installed and can be set up in your environment, but CHIME I would say is a bit more advanced. So a quick summary before I end with this section is that metagenomes refer to the host and associated microbes in a given ecological niche. Metagenomics has provided another lens to observe microbial diversity, functioning and phenotyping. The metagenomic studies can be performed with single gene or whole genomics. Likewise, we have meta-transcriptomics or meta-proteomics. We have two platforms, Mothur and CHIME, and are two important platforms. And here is a small cartoon where I am just trying to tell you that we really cannot ignore microbes, that we need to take care of all microbes around us for our well-being. So with this one, we come to an end of this section. I'll see you again after a while. Thank you very much.